So good afternoon. We're on Stanford Daylight Time, which means the hour starts 5 minutes after the hour. But it is 5 minutes after the hour, and so we'll get started. My name is Hank Greely. I'm here wearing two hats, although you may be saying to yourself, I don't see any hats at all. But in one hat, I am the director of Stanford Center for Law and the Biosciences, one of the sponsors of this meeting, this program. And in that role, I'm very happy to welcome all of you here. If any of you is not, foolishly, is not yet on the mailing list for the Stanford Center for Law and the Biosciences, let us know, and we'll make sure that you get notices of all of our upcoming events. Not many left this academic year, but we'll be back next academic year with a full slate of events. We're a Stanford Law School center that is dedicated to encouraging education, research, and teaching on the intersection of law and the biosciences. But now, switching hats, I also want to welcome all the people here who at some point, at some place, at some time in their lives have been elected to Phi Beta Kappa. Because this meeting is also co-sponsored by Beta of California, which is the Stanford chapter of Phi Beta Kappa. For sins that I've not yet quite identified, I am now the president of Beta of California. And one of the funny things about being the president of a Phi Beta Kappa chapter, most of what you do with Phi Beta Kappa is as soon as you elect your members, you send them away. Because 90% of them get elected, 80% of them get elected at just before graduation. So there's an initiation on the Friday before graduation. So the membership is here for 2 days, and then they're gone from Stanford for the most part. And those 2 days, they're busy with other things. One of the things I would like to see us do as Beta of California is to try to get more activity during the year from people who are Phi Beta Kappa, not just at Stanford, but any place else. And I do know that between the faculty, the staff, the graduate students, and the postdocs and others, there are lots of people who were elected to Phi Beta Kappa one place or another. We're looking for ways to try to make Phi Beta Kappa more of an actual organization and less of a 1-2 hour ceremony at which people get certificates handed out to them and their backs patted. So if you are a Phi Beta Kappa member and you got any thoughts about that, let me know. Or let our two officers know, Melissa Stevenson, who is our secretary, and Mark Tyndall, who is our treasurer, both sitting in the back. So I think that's all the miscellaneous announcement I need to do. I'm really happy that you all showed up, and I'm really happy that our speakers showed up, because I think this is just because it would be a real drag if none of the speakers showed up, especially for you guys, because you just hear me for an hour and a half. This is a really interesting topic, and it's an interesting topic that's come up now twice in two slightly different contexts in the space of just the last few months. The topic, as we're talking about it today, is the issue of human germline genetic or genomic engineering, which means changing the base pair sequence, the genomic sequence of humans, in a way that you intend or expect will then be passed down to any descendants that they have. And those descendants, in turn, may pass it down to any descendants they have, and so on until the end of our species. Germline engineering is something that people have talked about in science fiction and movies and TV and books and comic books for a long time. It has never seemed particularly realistic as a medical intervention until recently, because the cost, the efficiency, and most importantly, the accuracy and safety of making modifications, specific modifications to the human genome in embryos or in sperm or in eggs has been dauntingly too expensive, too inefficient, and way too risky. Now, I think it's still way too risky, but we've now seen the rise of new techniques that make it plausible, much less risky. How much less risky, we still don't know, but make it plausible that we can make changes in the genomes of either parents or embryos that will then be passed down to any descendants. And interestingly, it's come up in two different contexts just in the last few months. One is mitochondrial DNA. So all of us inherit our mitochondrial DNA from our mother. The mitochondria, you may remember from high school biology, is the energy powerhouse of the cell that's shaped like a kidney bean. Actually, I had no idea what a kidney bean was shaped like until I saw a picture of a mitochondrion. We get that from our mothers, and we don't get any contribution from our fathers. And if a woman has a disease that's caused by a defect in her mitochondrial DNA, that will be passed down to any children she may have. There's now been discussion in the UK, has actually in Parliament passed legislation allowing mitochondrial transfer, to take mitochondria from one woman's eggs and move it into another woman's eggs to try to deal with that. That would then get passed down to all of her children and all of her daughter's children. It wouldn't get passed down to her son's children, but all of her daughter's children and all of her daughter's daughter's children and so on. The other method is more wide-ranging. The mitochondria has its own DNA, but it's only about 170,000 base pairs. And although 170,000 sounds like a big number, compared to the roughly 6.8 billion base pairs in the nucleus of cells, it's not a very big number. New methods of doing genomic engineering on nuclear DNA have been developed in recent years. One of the main developers, Jennifer Doudna, is at the University of California at Berkeley, that have transformed genome engineering by making it faster, cheaper, and more accurate, by somewhere in the vicinity of an order of magnitude. I think this is like the transformation in America that took place when we moved from really expensive automobiles to the Model T. There were cars before the Model T, but they were uncommon, they were expensive, they weren't making a huge impact on society. There are cars after the Model T. Nobody drives a Model T anymore, but the Model T was the inflection point, the point where it changed and it became part of everyday life in a way that, not necessarily everyday life, but every scientist's daily life or every molecular biologist's daily life, I think CRISPR-Cas9, to give its full name, is at the inflection point that means this is going to be more common. So there's been a fair amount of discussion, including a couple of articles, one in Nature, one in Science, calling for somewhat different moratoria on human germline gene therapy. We're here because, although it's not, I think almost no one would say the CRISPR-Cas9 part is ready to be used. It's a lot more plausible than it ever was before, and it may happen soon. So the lineup we have today, and I was just telling my speakers, I don't believe in long introductions, so about 6 words per speaker is likely to do it. We'll actually start with Lynn Westphal here, who is a professor of OBGYN at Stanford, followed by Chris Scott, who is at the Stanford Center for Biomedical Ethics and the director of Stanford's program in Stem Cells and Society, followed by Marcy Darnovsky, who is the director of the Center for Genetics and Society, located in the East Bay someplace.
Berkeley.
Don't say that word. Followed by Paul, and Paul, I've seen your name. I can spell your name. Knoepfler?
It's a silent K, Knoepfler.
Knoepfler. Knoepfler, right?
Designed to torture you.
Right. Paul Knoepfler, who is a scientist at UC Davis Medical School. And then I'll talk a little, and our hope is that we will all be prompt enough and taciturn enough. It's a hope, it may not be a realistic hope, that we'll leave plenty of time for discussion among us, but more importantly, discussion with you, discussion from the audience. So I'll shut up for now and turn things over to Marcy. Not to Marcy, to Lynn.
Well, thank you for inviting me. Hank and I Professor Greely and I first
Dr. Westphal.
On a flight from Sun Valley here. We were at a conference and we were discussing, some of our common interests. And I think that was when I first got involved because of our common interests in these areas.
That's where this meeting was born, in a very small plane.
In a very small plane from Sun Valley. I'm a reproductive endocrinologist, and I take care of a lot of the patients who probably would be accessing these treatments. And many of these techniques would be part of an IVF cycle, which means that the women would go through about 2 weeks of ovarian stimulation, doing daily injections of medication, then having their eggs removed, fertilized, then will have done whatever needs to be done, and then the embryos would be transferred back. So historically, almost 20 years ago, there was a technique that was used for infertile women who seemed to have eggs that just weren't functioning well. So this was a technique called cytoplasmic transfer, and they would take cytoplasm from a donor egg and put it into the recipient mother's egg. And in doing this, they would take a small amount of mitochondria and put it into the recipient's egg. There were about 30 pregnancies from this technique, and it was stopped because of a lot of safety concerns. There were a couple of fetuses that were abnormal. They had Turner syndrome, missing an X chromosome, and so the FDA banned the technique in the U.S., and that was probably around 1999, maybe, that they banned it.
I was thinking around 2001.
Yeah, so
around the turn of the millennium.
yeah, so it hasn't been available in the U.S., but interestingly, I do have patients who still ask me if we can do things like this. As Professor Greely said, there are a lot of inherited mitochondrial diseases, and they were first described in 1988. Research has identified quite a few of these. There are probably over 250 known or identified mitochondrial diseases. And the clinical symptoms are very diverse and often affect vital organs that rely heavily on energy production, such as brain, heart, liver, muscle. It's estimated there are about 1 in 4,000 people who are diagnosed with mitochondrial disease, but because the symptoms are so varied, it could be higher. Mitochondrial DNA is maternally inherited, so that's why it's different than inheritance from the nucleus. And the fertilized egg contains several 100,000 copies of mitochondrial DNA, and these get distributed randomly into the embryo. So each cell contains multiple copies of mitochondrial DNA, and then the percentage that have mutations and where they are in the body will determine the symptoms that appear. So, for example, you could have a woman who has hearing loss related to this, but she could have a child that has severe symptoms, seizures, other problems, or she could have a completely healthy child, just depending on how the mitochondria are distributed to that child. So the new techniques related to mitochondrial transfer are more advanced than those cytoplasmic transfers that people did years ago. And it's different because it proposes to replace all mitochondria. There are a small number of women who have these mutations who I think would benefit from the way the current technology is being proposed. However, although there's no evidence that doing mitochondrial transfer can treat infertility, I think this is where a lot of patients and fertility experts think that it may go. So some people think that by doing this, you could rejuvenate eggs of older women and use that instead of going to donor egg. So a lot of older women, if they have poor egg quality, will need to use eggs from someone younger. And so the hope, if this technique did work, would be that perhaps it could make old eggs younger. The other area that's really exploding is the area of genetic screening. And as there are more mutations that are identified, there are more patients who are interested in testing so they don't pass on these diseases. So current recommendations by professional societies recommend genetic screening on all pregnant women, although the extent varies by organization. We currently in our clinic offer a panel to patients that test for more than 100 different genetic mutations. Our professional organization, the American College of OB/GYN, recommends testing everyone for cystic fibrosis because that's the most common mutation. And in that situation, if both parents are carriers, they would have a 25% chance of having a child affected with cystic fibrosis. So if they did become pregnant, you could test the fetus to see if it had the disease, and then the patients could potentially think about terminating the pregnancy. A lot of couples don't really want to have to be in that situation. So then we have couples who are coming in to test their embryos and doing pre-implantation genetic diagnosis before they get pregnant. So what they do is they go through a cycle, we fertilize the eggs. 5 days later, we can do biopsies of the embryos and test to see if they carry the disease. So this is a fairly common thing that's done for people who know that they carry a disease. And unfortunately, a lot of the women who are going through and testing for disease tend to be older, and we often will go through the whole process and they may not have any normal embryos to transfer. And so that's obviously very disappointing in someone who's gone through 2 weeks of treatment and great expense. And related to that, there's a lot of debate about which test should be offered with pre-implantation genetic testing. So most people agree that it seems reasonable to test for things like cystic fibrosis, where it's a life-threatening disease for the child. But there are other things that we do test for. So people who know that they have an increased chance of having cancer because they carry the BRCA mutation, for example, we can do testing for that. Then there are other situations where you can do testing to see if the fetus would be an HLA match for a sick sibling. There are countries that do not allow that. In the U.S., that is something that is available, and we've had patients who have had a child who needed a bone marrow transplant or wanted cord blood because they had a serious disease. And then PGD sometimes is just used for social situations. We have patients who come in who have 2 boys and want a girl, or 2 girls that want a boy. So, we're already dealing with a lot of ethical issues in our clinic with how people are using some of these technologies. So the ability to correct mutations in the embryo would greatly increase the number of normal embryos that we have and would increase the pregnancy rate for these patients. And I do envision that a lot of patients would really be pushing to get this technology as quickly as possible. And so I think it's really important on our side that this whole area with gene editing that we do as safely and as ethically as possible.
Thank you, Lynn. Chris.
Great. Paul, could you pass me the little, the control, actually it's the keyboard, I think.
Oh.
I only have to punch it once, but can't. So, thanks, Lynn, for the introduction, and thank you, Hank, for inviting me. As I'm standing in for a scientist today, I'm going to give you a little bit of science, and nothing is more dangerous than putting some science in the hands of a ethics, law, and policy scholar. So I know there are a couple of scientists in the audience, so if you see me misstep, raise your hand and give me the correction I need. So this is a slide from the Doudna paper, schematically showing the CRISPR technology. And CRISPR stands for clusters of regularly interspaced short palindromic repeats. Say that as fast as you can 7 times, and we'll test you at the end of the class. So the diagram shows a double-stranded piece of DNA bounded by a couple of scissors there. And CRISPR actually is a natural product of an acquired immune system in bacteria that allows short bits of viral DNA to be incorporated into the bacteria's genome. It functions to protect them from subsequent infections, so it's really a pretty nifty system. The viral bits get transcribed into RNA. RNA then pairs with something called Cas, which is called the CRISPR-associated protein, and together that group of molecules interfere against new viral infections. Basically what it does is it turns the viral infection against the virus itself. And that's why it's pretty cool. So Jennifer Doudna and her collaborator, Emmanuel Charpentier, uncovered the function of Cas9. And what they did in, I think this was 2012, is they found that there are two RNAs that recruit the Cas9 protein. The Cas9 protein is the thing that actually cuts the DNA. It cuts it in both strands. So it's a double cut. And it generates sequences that match something called the guide DNA. So you wonder how the molecule actually gets to the DNA. It's basically a base pairing phenomenon where the RNA matches up with the DNA, and it's at that specific site where the cuts occur. So it works pretty much like this. Cas cuts the DNA. The two pieces of mRNAs form a complex with Cas, and then it activates the system, and then it repairs. So there are two types of repairs that happen. One is after the cuts, the ends simply join together. That's called non-homologous end joining. That happens sometimes with a mutation with a little bit of extra DNA, or it takes out a piece of DNA. That's commonly called knocking out. Then you can also bring in a piece of DNA in a homologous fashion, so you're actually introducing a new piece of DNA into the genome. That's one of the powerful effects of the technology, and that's called homologous recombination. So, or also knock-in. So we've got knocking out genes and knocking in genes. So let me go to the next slide here. So here's an article that appeared about 2 weeks ago, was it, Hank?
Yeah, a little over 2.
2 weeks. I can't.
Late April.
Okay, about 2 weeks ago, that showed this proof of concept in human cells, and this is what's generated all the controversy and is really behind why we're gathered here today. It was in a little-known journal, at least little known to me, called Protein and Cell. These authors in China had originally submitted this paper to Nature and Science, the publications that had the two commentaries that Hank talked about. And for reasons that are not quite clear to everyone, we'll probably get into this discussion, the paper was rejected at both of those journals, accepted here, and published with a 48-hour turnaround. So the authors did this in a human embryo that was triploid. So it was a non-viable embryo. That's an important point to remember. They found three things. One, it was a very low efficiency of editing. So less than 50% of the embryos were actually edited. They found mosaicism, which means that the embryos started to divide before the corrections were complete. So some cells were edited and some cells were not. And most importantly, they found lack of specificity. So one of the important advantages of these new types of precision genome technologies is an improvement over the old viral-mediated gene delivery systems of the 1990s. In this case, they found significant numbers of off-target mutations. In other words, the gene that they were trying to correct was inserted in places where they weren't targeting. So about 4 days later, the editor of the journal wrote an editorial that addressed some of the public concern and ethical concern that came out when this paper was published. And I'm just including one quote as a way to kind of get us into the conversation that I think is really interesting. I'd strongly urge you to read this. I think it's one of the most fascinating parts of this entire story, really. And he said, this experiment should not be viewed as an endorsement of this practice, nor an encouragement of similar attempts, but rather the sounding of an alarm to draw attention to the urgent need to rein in applications of gene editing technologies, especially in the human genome cells, or human germ cells or embryos. So here in a few words, it's almost as if this journal and the editor were publishing this paper as a kind of public service, which I think is pretty interesting. So we can have the lights back on. In conclusion, some of the interesting ethical issues that this raises for me are that the guidelines and recommendations that are coming out of the Science and Nature papers are split on an important issue. Waiting for the machine to stop, that's funny. And that's whether the research should proceed in vitro, in other words, not for making babies, but rather only in the laboratory to determine whether or not the technology is safe and efficient and efficacious. The International Society for Stem Cell Research has come out in favor of those experiments that would go in the laboratory. And Hank, your editorial also seemed to suggest.
One of only 18 authors, but it called for a moratorium on making babies this way, but did not call for a moratorium on research.
On the research. And then there are other organizations that have gone a different way, saying that there should be a moratorium or banning the research both in the laboratory and for clinical applications. So that's an interesting, I think, public policy split. Also, another interesting thing we could talk about is the reasons and rationale for the Protein in Cell publication. What was behind these authors and the journalists and the journal editors that actually published the paper? And it raises, I think, other deeper questions about why these types of papers are published in the first place. So we can posit why Nature and Science turned the article down, but some things that I've been thinking about with collaborators are whether or not this research raised the bar and gave us new information that we didn't already have in studies that didn't use embryos. So was this really a transformational sort of paper, or was it rather a gee whiz thing that just showed that it could be done, the technical feat? And what should journal editors and reviewers do when they're considering something like this? The splash factor, something that's never been done before, but maybe not technically very high on the scale, or something that actually moves the field forward because it's new information? That's a question I've been thinking about. The final thing that comes to mind in light of the reasons for publishing is if we would decide that some laboratory research would be justified going forward with this technology, what would that research look like? And I think that's where the discussion is headed now, where scientists and ethicists and policymakers in meetings like this debate and discuss what types of projects in labs at Stanford and Davis and other places might actually move the field forward in essential questions of human development. Fertility is certainly one place, it seems to me, that an in vitro application of this would be pretty important, but something to think about. Thanks, Hank.
Thank you, Chris. Marcy.
Okay, well, thank you, Hank, for inviting me, and thanks to all of you and all of you. For me, the central question that we need to be asking is whether we should proceed with creating genetically modified human beings. That's the one that I think is important, and that's why there's all the fuss here. So from that point of view, it's really unsettling for me, instructive and unsettling for me to hear, Lynn, you talking about it as a simple extension of IVF techniques. I don't see it that way. Yeah, so we'll talk about that more. Yeah. So I'm not going to address the mitochondrial issue very much, except to note that in the UK and in other countries, germline modification in human beings is against the law. That was settled some time ago. And the reason the UK Parliament had to vote on this particular procedure was it does modify the germline, and they had to carve out an exception in that rule. Because it is a germline procedure, we've been following it pretty closely at the Center for Genetics and Society for some time, and have real concerns about its safety and about the process in the UK and so on. But what I'm going to be talking about really is the questions around human germline modification, where we're at now, where we should be going or not going, And Hank, the instructions were to give some thoughts about ethics. So I thought I would start with some thoughts about what we mean by ethics. And the first thing I want to say about that is that I think we have to be careful that we do not collapse ethics to just mean safety or something being safe enough. I was really surprised I was on a PBS talk show last week or the week before with a Nobel Prize-winning geneticist who said flatly, if we can improve the safety, he was talking about CRISPR gene editing, if we can improve the safety, then that takes care of the ethical concerns. I don't see it that way at all. Safety, of course, is huge, paramount, but it's hardly the whole picture. The other thing that I think we have to be careful about when we're talking about ethics is not to collapse it just to questions of individual consent. Like safety, informed consent is important. When we're talking about modifying the genes that we pass down to our children that would affect every cell in the bodies of those children and their descendants, informed consent is a bit of a challenge, even if you think parents are okay to make those decisions for their children. There's no precedent for them making those kinds of decisions about their grandchildren and future generations. But that aside, to think about ethics just as consent is just wildly insufficient when it comes to assessing the prospect of genetically modified human beings. Some people have called human germline modification, or actually germline modification in general, a species-altering technology. And I think it's also pretty likely to be a society-altering technology. And that's why I think we need a much broader ethical focus. We really need to take a careful look at the social dynamics that introducing human germline modification would likely set in motion. And that includes the commercial dynamics that it would set in motion. Those are not the kind of risks that are easily quantified, but they are nonetheless very real. And I think we have to really anticipate them very carefully and thoughtfully. When you think about that, the worst case scenarios are actually pretty horrific. And these are the worst case scenarios, and we have to assess how likely they are. So what are they? A genetic arms race between nations, within countries, a world in which affluent parents purchase the latest set of upgrades for their offspring, leading to the emergence of genetic haves and have-nots. This is the scenario that a Princeton molecular biologist called the gen-rich and the naturals. And part of his speculation was writing ad copy for a fertility clinic of the future. And he called their services organic enhancement. And the ad copy went, keep in mind, you must act before you get pregnant. Don't be sorry after she's born. This really is a once-in-a-lifetime opportunity for your child to be. So this is the kind of vision of the future that this guy actually seemed to be endorsing. He wasn't doing this as in a cautionary mode. So many people, of course, don't want to go there. They would like to avoid that kind of scenario that could be caused by the emergence of genetic castes, the idea that wealthy people will buy these upgrades for their children. They don't want to embrace that, but they do want to embrace the idea that we should be using germline gene editing for medical purposes. And of course, we all want healthy babies, and we want to help people have healthy babies. But here's a really key point. As you referred to, Lynn, we don't need germline modification in most cases, in almost all cases really, to prevent the transmission of inheritable diseases because of the embryo screening technique that Lynn described. Pre-implantation genetic diagnosis is what it's known as, PGD. And that can provide an unaffected, a healthy and fully genetically related child for a couple that is at risk of passing on a genetic disease. And it would also eliminate that mutation from the family lineage. And it's not risk-free, and it's not ethics-free, as you mentioned, especially from a disability rights point of view. It's far less socially and ethically fraught and far safer than mucking around in the human germline, to use the technical term. So then you have to ask, okay, if you can use PGD for preventing the cystic fibrosis, the Tay-Sachs kinds of diseases, why not use these new gene editing techniques to engineer people with what you might call medical enhancements? So, for example, to give a future child, swap in a gene variant that would provide immune resistance to HIV. But think about where that takes us. The line between medical and therapeutic traits on the one hand and enhancement or cosmetic traits on the other hand is impossibly blurry. That's inherent in the logic of it. So that would leave us with no way to define a meaningful line between different purposes, and therefore no possibility of really establishing or let alone holding a policy line. And when you think again about the real world of social dynamics, of commercial dynamics, you're a couple, you've already invested in the genetic upgrade to give your child immunity to AIDS. Well, then, why not throw in a few dozen genetic variants that we think might boost her IQ too? You know, no additional cost, no additional risk. Then you have the child, she grows up, and she gets her high SAT scores, and how do we know? Is it because of the genetic upgrade, or is it because of the high-end private school? Wouldn't that kind of situation make it pretty likely that we would start to think about some people really being genetically superior, biologically superior to others? And then what? So now some observers, and Hank is one of these, point out correctly, of course, that these kind of traits that would make our children qualify as designer babies, the higher intelligence, the perfect pitch, and so on, that these are extraordinarily complex. They're multifactorial, even just in terms of genes. They interact constantly with the rest of the biological and social environment and so on, and that's all true. And for some people, the conclusion, therefore, is we don't have to worry. And I have to say, I do take some comfort in the thought that these designer baby scenarios may remain beyond technical reach. But then I think about the glow-in-the-dark monkeys, about the transgenic mice that run the mazes faster and more accurately than their natural counterparts. And I think about the commercial forces and competitive dynamics that might make parents in affluent communities like this one feel that they were being irresponsible to deny their child the best start in life, the once-in-a-lifetime opportunity. So these questions, all this kind of thinking about, is there a medical argument? What would be the social consequences? These are not new. They've been under discussion for decades. And back in the 1990s and early 2000s, these questions were actually hashed out in a lot of places around the world, and a lot of jurisdictions in more than 40 countries decided that this was a no-brainer, really, for most of them. It was not going to be done. They passed laws. Those laws are still on the books that encourage using all kinds of gene transfer techniques for medical purposes to help people who are sick, and prohibit using them to modify the human germline. There are also this is the law. Not these 40 countries. These are scientifically advanced countries, the ones with the biotech sectors. And there are also a couple of international documents, including a Council of Europe treaty that prohibits human germline modification, just puts it off limits. These laws and these treaties, they draw a very clear and very practical line between somatic gene transfer, as it's called, and germline interventions. They see one as an effort to treat people who are sick, and that these techniques should be strongly supported, and they see germline as an effort to breed superior people, and we shouldn't go there. And I very much in sympathy with that point of view. So the medical argument for human germline modification is tenuous because we have other techniques to prevent inherited disease. And the temptation to try to enhance future generations, I believe, is profoundly dangerous. Down that road, our scientific achievements are all too likely to become not instruments of enlightenment and emancipation, but instead to become mechanisms for exacerbating inequality. Down that road, our desire to improve the human condition, which I think we all share, leads us away from the social and political changes that we know are needed to provide that we need to provide people with clean air, good food, basic health care. And instead, our wonderful technical and scientific capacities all too easily wind up actually fostering some of the least attractive tendencies of our time. and I would number among those techno-scientific curiosity that is unbounded by care for social consequences, also hopes for our children that are wrought into consumerism, and another I'll mention is a deep denial of our own mortality. So I think it's something not to be done. And just one last observation. We often hear that human germline modification is inevitable, and I think that's just not true. Societies make decisions all the time about the rules of the road, which ones they're going to enforce, and they change. Human societies haven't always outlawed slavery. Human societies haven't always outlawed murder. And in fact, in 2015, there's still murder in the world, and there is still slavery in the world. But we would be living in a very different world if laws against murder and laws against slavery didn't exist. And we're likely to find ourselves in a very different world depending on what we decide about how to use the powerful genetic tools that are now being refined.
Thank you, Marcy. Paul.
Thanks for the invitation to come and speak. I'm going to give a little bit of a different perspective. I'm more of a bench scientist, so I'm a person who actually goes and tries to make these genetic modifications, and that's something that I've done in different forms for the last almost couple dozen years. So I'm going to try to give you that perspective, and I would respectfully disagree with Marcy in that I have a different view in that I think that human genetic modification is inevitable. I really do. And part of that stems from just in the last few months, all the interactions that I've had with other scientists and biomedical scientists, physicians, and the level of sort of attachment and excitement about CRISPR and about gene editing is really something that I've never seen before in my career. I don't know if any of you are old enough to remember before we had something called PCR. PCR is a technique where we can make Xerox copies of DNA almost in an infinite kind of manner. And before that, we were really stymied in science in a lot of ways. I'm a molecular and cell biologist and developmental biologist. And as soon as I started grad school, we had PCR, and it was something that we all relied upon. But before that, we were really stuck in a lot of ways of what we could do. And I think PCR totally revolutionized my field of science. And I think CRISPR and other related gene editing technologies are poised to do the same thing. And so I kind of view where we're at right now with CRISPR-Cas9 and the power of that gene editing as sort of a good news, bad news kind of situation. And I think on the one hand, scientists have kind of lashed on with this super excitement, energy, onto this super powerful technology. And what I would tell you is that's the good news and the bad news. So on the one hand, that's good news because there's a lot of really amazing science that we can do with that technology. The energy that I see with scientists when they talk, their face lights up when they talk about CRISPR-Cas9. Literally, they're practically in love with it. And so I think on the one hand, that's the good news. We can do tons of great science with this technology. On the other hand, I think there's so much enthusiasm and there's this momentum going forward amongst thousands of scientists all around the world. And because CRISPR-Cas9 is something that you can literally go on this computer and just click a few clicks and order the materials to do it, there's nothing, special about it in that regard. I really think it's in the hands of so many scientists, so many physician scientists around the world that I just can't imagine that someone won't take the plunge and go for it. I'm with Marcy that I really think this technology, there's a lot of things about it to be worried about, but I guess I'm less confident that it won't, someone won't go ahead and try to genetically modify germ cells and embryos with the intent to actually make people. So I'm a scientist, but I also have a blog where I try to engage in advocacy and policy discussions. And so I weighed in on the mitochondrial transfer issue that Hank alluded to earlier. And that was something that I'm concerned about. And then as I was sort of going through that in a policy kind of perspective, kind of putting on my policy hat, I was starting to hear more and more rumors about CRISPR-Cas9. And people, this was kind of late last year, people were really worried that there were going to be papers coming out like this one that Chris showed, the one from China, reporting editing of human embryos. And so that was something I started really getting engaged with. And So I've actually come out on my blog with a plan for how to try to grapple with the emergence of this new super powerful technology. And I think one of the key factors is really oversight and training and bioethics training for this whole community of scientists out there, because they've got the technology, they're sitting there, they've got it in their lab, but they may not really think about the ethical issues, the legal issues. They might not think about transgenerational issues, about things affecting grandchildren and great-grandchildren and things like that. And so I really think that kind of educational effort will be instrumental. Again, I don't think it's going to reach everyone. I don't think it's going to stop some people from going ahead with things they probably shouldn't do. But I think it's better than doing nothing. I really feel like we need to allow the in vitro research to proceed, to go ahead and do research in the lab. But I do recognize that if we start editing embryos like was done in this paper from China, at some point that might be a stepping stone towards actually making a genetically modified person. So I know that there are definitely risks involved with that. So I think I'll just end at that point and be happy later to take questions.
Great. Thanks, Paul. So my turn. I'm the lawyer, so I'll start with the two words that a lawyer should answer any question with.
It depends.
It depends, because it always depends. And I want to make 5 quick comments before we open it up to more general discussion. First, we talk about germline gene modification, but I think it's important that we think through exactly what we mean in this context, because what you mean by germline modification is part of what it depends on. All of us are the result, to some extent, of unintentional germline modification, because mutations happen every time a cell divides, and none of us is born with exactly the same DNA sequence that our parents' eggs and sperm gave us. Their eggs and sperm differed from the eggs and sperm they got from their ancestors, and what they passed on to us was mutated, usually in small and unimportant ways, though not always. And this goes on throughout our lifetimes. The skin cells on this palm are different in their DNA sequence from the skin cells on this palm because they are about approximately 63 years separated from when they were one cell. Every time they divide, there's a little bit of a change. So the germline doesn't stay inviolate. It doesn't stay pure. It's always being affected some, though randomly, and not with human direction. Whether that's a good thing or a bad thing is a different issue. Second, with respect to the germline modification, if what you are doing is taking a version of a gene, an allele, one gene version, that is known to be disease-causing, like a 182 del AG mutation in BRCA1, which puts women at much higher risk for breast and ovarian cancer, and you're replacing it with the average version that most people in the world have, you're changing the germline for that particular embryo, baby, etc. Are you changing the human species germline? Not really. You're just changing the incidence, the prevalence of different alleles. And we change allele prevalence all the time. Civilization has changed the prevalence of different alleles. Agriculture leads to some genetic variations being favored over others. Diseases lead to some genetic variations being favored over others. The invention, the discovery by Banting and Best of insulin in the 1920s means that there are more people alive today who are genetically predisposed to get type 1 diabetes than otherwise would be the case, because now they can survive long enough to actually reproduce. So it's a little tricky. If you're moving chimpanzee alleles into humans, you're changing the germline genome of the human species. If you're moving them from one human to another human, you're changing the relative frequencies, but you're not really changing alleles, gene types that are found in humans. Second point, safety. I think this would be criminally crazy for somebody to try to make a baby this way right now, in part because of the results of that Chinese experiment. Yes, it works kind of, but not very much and very, very poorly. If you want to experiment on, again, I'll say in this case, 62 years old currently, although embryonically my cells are 63 years old, 62-year-old man, and change some of my cells because I've given informed consent to try to get a disease fixed, if it goes catastrophically wrong, okay, it's sad. Some people would be unhappy. Some might not be. But it's one person who is a competent adult, made a decision to go ahead and risk it. If you try to do something to make a baby, first, that baby never consented to it. And second, you may be inflicting 70, 80, 90 years of suffering on that child who never gave consent. Making babies is serious business, even though most of the time I think people do it without giving it sufficient thought. But it is a serious business, and the safety has to be really beyond reproach. And we are, I think, at least one and probably several decades away from being able to say, this is safe enough. This being the CRISPR-Cas9, I think the mitochondrial DNA may be a lot closer, but this is safe enough to try to make babies with. Third point, safety is always a relative thing. If I came up with a drug that instantly cured half of people with metastatic pancreatic cancer, one of the worst diagnoses you can get, and instantly and painlessly killed the other half, that's a wonderfully safe and effective drug in the context of metastatic pancreatic cancer. If you use that drug for teenage acne and it cures half of them and kills the other half, not such a good drug. You have to weigh safety in the context of benefits. And here I agree with Marcy that the benefits from human germline modification are relatively small. Actually, I think quite small. Most genetic diseases can be prevented, if you want to do it, if you want to go down that route, by either pre-implantation genetic diagnosis, which Lynn described, which sounds like science fiction, but has been around for 25 years. About 4,000 babies were born last year in the U.S. who'd had one or a few of their cells plucked from their embryos and tested, or through fetal testing followed by termination. So there are other ways around it for almost everybody, but it's only almost everybody. I can think of 3 examples where you can't use those selection techniques to get a healthy, or at least not known to be unhealthy in this particular way, because no one can guarantee you a healthy child, but we can tell you that this child will not have this disease when it's still your genetic child. One example is people who have what we call an autosomal dominant disease that takes only one bad copy of the gene to cause the disease. Huntington's disease is a good example of this. But they actually have 2 bad copies. One is enough to get the disease. They've got 2 bad copies. If they want to have kids, quote, of their own, close quote, a phrase I don't like, because I think adoptive parents have their own kids too, but if they want to have genetic children, they can't use their sperm or eggs to make genetic children that won't have Huntington's disease, because that's the only allele they have to pass on. Now, how many people are homozygous, have 2 disease copies of an autosomal dominant disease, who live long enough and are healthy enough to want to have kids? Probably not very many, but I can't say 0. Similarly, if a couple both have the same autosomal recessive disease, so think cystic fibrosis, Tay-Sachs disease, Tay-Sachs won't live long enough, but sickle cell disease, beta thalassemia, etc. If 2 people with CF marry and want to have kids, all of their children will have CF because they only have disease alleles to give. Each one can only give their child a disease allele, so that child will necessarily get 2 disease alleles, and if it's a sufficiently penetrant, sufficiently powerful genetic disease, they'll have the disease. If they want to have a genetic kid, they need to do something else. The third example is the mitochondrial diseases. If a woman with a mitochondrial disease wants to have a child, she needs to somehow get different mitochondrial DNA. Now, how many people with CF marry other people with CF and are healthy enough and decide they want to have kids? Probably not very many, but probably not 0. How many people with mitochondrial disease, and Lynn has estimated the incidence of, shall we say, true or substantially documented mitochondrial disease at around 1 in 4,000, maybe less, maybe more common. Not very many, but not 0. So there's some benefit potentially for those people. Everybody else can basically reach the same result using something that is much less risky, either PGD or fetal testing. Third point, and I think this is where Marcy and I most disagree, other than Berkeley, Stanford. there are there are two types of people in the world, the kind that say there are two types of people in the world and the kind that don't. That's a joke. I think there's a fundamental viewing of the world difference here in whether you want to be an absolutist or whether you want to try to regulate. So Marcy says, I think, if I were confident that it would be limited only to really important medical need to relieve human suffering and didn't go beyond that, I might be okay with it. Is that fair?
And I don't think there's a way to draw the line
Right.
there.
You don't think there's a way to draw the line there. And so you say, let's draw the line and say, don't step 1 foot over this line ever. And you poor folks who actually need this to have kids that are genetic children, sorry, you're out of luck. My view is, let's regulate it and say, you don't want it used for enhancement? Ban use for enhancement. You don't want it used for fertility? Ban use for fertility. Now, there is no perfect line there, just as there is no perfect line that determines when somebody is should be old enough to vote or old enough to drink. We say 18 or 21, not because they're perfect lines, but because they work, we think, well enough. There will be no perfect regulatory scheme, but I think there can be a good regulatory scheme. And note, by the way, Marcy's right, banning murder doesn't make murder completely disappear, although it makes it a lot less common, we think. If you ban it entirely, that doesn't mean it won't happen. It means it will happen less. Will it happen more or less if it's entirely banned than if it's regulated and limited to only a few situations? I'm going to guess on the regulatory side of that, but it is an open empirical question. Last point. How are we going to decide all this? There are about 45, 50 of us in this room. We can take a vote. Doesn't mean very much. Some of us are still of an age where we might be thinking of having kids of our own, as opposed to grandkids or great-grandkids. We can vote with our feet, so to speak, or vote with our gonads and make decisions about how we want to have our kids. How will society make this decision? And ultimately here, I believe that we should do it with the worst form of government ever invented, except for all the others that have been tried from time to time in a democratic way. And if, in fact, Germany wants to ban it, or South Dakota wants to ban it, but Singapore wants to allow it, or California wants to allow it, I think so be it. I don't think science and medicine both have to live within their societies. Let's have a full, robust, and open debate, and let the democratic chips fall where they may. So we can start now, moving beyond this table, to a full, robust, and democratic debate, or at least comments from the audience. I should warn you, though, anything you say can and will be used against you, because that little device up there is videotaping all of this. So if you choose not to have your comments spread over the World Wide Web, where we intend to place this as soon as it gets edited, which will be sometime in the next 30 years, next week, we hope, don't say anything, at least not during the regular session. If you are willing to be brave and be recorded and go on record, now is your time. We're open for questions. Yeah.
It's the opposite direction, but Michael Crichton has this book called Next.
Yeah.
Some communities so how close are we scientifically? Does this progress supposed to be doing large-scale screening? So I want to figure out what genes are responsible for language. So I introduce 1,000 genes to 1,000 different software limits.
Yes.
Yeah, I think that kind of thing, that would be enabled by this, but that kind of thing is already going on already with existing technology. And even in some areas related to human beings, probably the largest genomic sequence company in the world, BGI, which is located in China, has a project going on. I don't remember the exact name, if it's called Project Genius or Intelligence or something like that.
Something kind of off-putting.
Yeah, something like that, where they've collected DNA samples from, I believe, 1,500 or 1,700 of the smartest people in the world. And what they're trying to do is define genetic variants that are associated with the intelligence in these people. And so that's sort of, an example actually in the human context where, I guess one could say that's sort of a knowledge-based experiment. They want to advance knowledge. But on the other hand, once we say they identify 12, alleles associated with intelligence, the next step may be, well, let's try to do something, with that information, not just learn from it. So,
by the way, there's a nice article about that effort by Michael Spector in the New Yorker, I think last year. Spector's a good writer. It's
and
a
there's
good
also
read.
a documentary about BGI called DNA Dreams, in which the very young, very smart scientists who are doing this work are very explicit about saying they would like this to be used for germline modification.
And I guess the other point I would make is BGI has also, in the last year or so, gotten very involved in animal cloning. And so there's sort of and that's not the only example where we have sort of this coming together of genomic and cloning technologies. And the cloning that they're doing right now is for livestock and things like that. But it just makes me a little bit uncomfortable as a scientist when we start seeing cloning in the same lab where there's, sequencing, genetic modification, things like that all going on together.
Well, just lest we, start being tempted to be finger pointing only at China, the what is it, cognitive, whatever that project is called,
Right.
is being made possible by an American.
Right, it's an international project, and I believe most of the sequencing is just being done in China, but it's definitely a global kind of thing, and many of the super intelligent people are just from all over the world.
Just going to say, that's kind of a logical experiment of the future that you might worry about. But the CRISPR technology is so powerful now that it's being used in large-scale experiments that could say, for example, screen for a drug. So by targeting thousands of genes with the CRISPR technology, because it's so fast, it's so easy, and it's so cheap, you can actually devise high-throughput screening for, let's say, a cancer drug, to look for those particular cells that would be perturbed that might induce resistance to a new drug. So, and that's happening all over. You can find publications throughout the literature that are scaling up the technology just past these small experiments to thousands and thousands of cells targeting every known gene that we have. So in the short term, that's kind of where it's going, and I think that kind of leads to the next question about, well, if we go there, where would we go if we were going to actually modify traits?
So, two things. One on the intelligence. I think the intelligence looks like it is so many genes and so much environment and so much chance that maybe someday we'll be able to say this embryo has a 55% chance of being in the top half and a 11% chance of being in the top 10%, but probably not much more than that. But that's an open empirical question. When you look for these genes, though, you learn things that might help you figure out diseases that cause seriously impaired intelligence. So there is potentially a medical reason to look for it. Second thing, I actually think, despite being an author on this, the one that called for a moratorium on the human germline, I think the most important area for CRISPR is not humans. It is what someone, and I wish I could remember who coined this, called CRISPR critters. What we can do to the rest of the biosphere using this technology is really pretty impressive. So a kid with a college, with an undergraduate BS and a couple $1,000 in a garage might be able to tinker around with mosquitoes to try to make a mosquito that couldn't carry successfully the malarial parasite. Could, in theory, release it and change all of the world's mosquitoes in ways that might be good, but maybe not. So I think the non-human stuff deserves a lot of attention, but probably not tonight. Other questions?
I guess the other thing I would just jump in and say that kid might decide to make a glow-in-the-dark mosquito just for the heck of it, and release
Easier to
that.
catch.
Or, right, easier to catch, easier to see at night instead of just going like that. So,
Yeah.
I'd like to take it back to the democracy
Yeah.
question, which in a way also fits in with the animal work, because a lot of people think that, or appear to think, that if you're working on animals, just go ahead and do it.
Yeah.
But I'm disturbed that the beginning of the debate in the last month or so seems
seems to have been, within the science community, seems to be held very tight. People talking about wanting to avoid government regulation, wanting to have professional regulation. Now, there's a place for professional regulation, I don't deny that. But I would like to see a much broader discussion, which means, of course, a certain amount of public education.
Right.
And similar things have come up with some of the genetic testing ethical issues, whereby if I find out something about my genome, do I tell my brother, who may well have similar issues? And this is sort of what's going on in a very broad sense when you talk about the human genetic inheritance, that it's not individualistic. However, decisions in practice do get made by individuals.
Or get made by individual countries and not by the world as a whole.
Right. And I think that there's an important gray area here that needs to be discussed, and I'd like to see the discussion move forward.
So the science piece had actually 4 recommendations, and one was a voluntary moratorium on making babies, on clinical use of germline modification, recognizing that in many jurisdictions it's illegal, including, according to the FDA in the United States, but if it's not illegal, you still shouldn't do it. Another recommendation was continued research to understand the strengths and weaknesses of the technique. A third recommendation was education sessions of scientists and non-scientists to help people understand what the issues were. And the fourth was calling for a big international meeting at which this would be discussed. The meeting, I believe, is going to happen later this year. Sadly, from my perspective, it's not going to follow. It looks like it will not be at Asilomar, which was the famous site of a meeting 40 years ago, exactly 40 years ago, on recombinant DNA. But I do think some of the scientists probably want to avoid outside regulation, just as they did at Asilomar. Asilomar ended up leading to NIH and HHS regulations on biosafety, but it did mean that Congress didn't pass statutes. So there are a lot of intermediates. I do think that getting the scientific discussion, which has spilled into the newspapers, going is the first step to subsequent public conversation. But it's the public conversation has to happen.
I was going to comment on that a little bit, and the fact, yes, it has started within the science community. In fact, it's been going sub rosa in the science community for a couple of years now. So it's interesting to see what was kind of talked out in committee meetings and other kind of, for instance, experiments now has reached public consciousness. That's the first point. The second thing that I've noticed is the fact that it's coming from the scientific community without a lot of, let's say, public consciousness is reflected in how the press has treated this. So some of the research that I do at Stanford is how the media responds to news like this. And over the past couple of weeks, there has been media coverage on this that has ranged from excellent to absolutely horrible. From the place we are engineering human babies, that the Chinese are engineering human babies, which is incorrect, that experiment there was done unethically. As far as I can tell, it was done completely ethically. Whether it was a matter of good public policy is the second question. And whether or not designer babies were on our doorstep, and as Hank says, probably not, and maybe not ever, and not anytime soon. So I think the function or the lesson maybe for this is that science needs to get quickly into the public sphere, and the best time to do that perhaps was 18 months or 2 years ago. And now we're kind of in the process of kind of looking back and saying, oh, how do we rein in all of this so that the discussion is kind of level-headed.
And even though I disagree with the Council for the Center for Genetics and Society on probably 80% of positions, I think they play a really important role in raising these arguments. And unlike some of the people who tend to be more opposed to some of these things, making better arguments and not just saying it's yucky or it's unnatural or whatever, that helps get public discussion going too.
I would point out that I agree. Democracy is the best bet we have, but democracy covers a lot of sins. So we don't really have much in our democratic society in the way of a sort of habit of mind or specific policy mechanisms to really grapple with not just questions about human germline modification, but all powerful technologies. And we, for better or for worse, have come to rely a lot on civil society organizations to raise some of the questions about, neonicotinoids or big dam or any powerful technology. And I think that's where what we're going to have to do with this question too. So I would like to see the involvement of all sorts of NGOs, civil society groups, labor unions, community groups, and I think that's what we need. And I think this is the kind of issue that is really poised. That the inflection point that you mentioned, I think it was you that mentioned it, Hank. We're also, I think, at a tipping point, an inflection point in public awareness of this issue, and that before long we're going to see it really spreading. And what we don't want is the misunderstandings, the misinformation, the partisan political divides. What we do want is a really robust conversation about the ethics, the social policy consequences, and just the societal futures that we might be facing, depending on the decision we make about whether we should pursue genetically modified humans.
I agree, but I also think it's important that it be appropriately informed by good science, which is never
Absolutely.
Perfect science, but good science.
Yeah.
Yeah.
I guess one question is...
You blew your chance, Chris.
Yeah, sorry, I had an idea, but
Paul first and then
I'll go
Chris.
second.
I guess I would say one question is, could it ever really be ready for prime time? And I'm not so sure, we can be definite about that. I think... Right, that's a good point. Right, and I think it can be really tricky to decide, what's an appropriate use. And I think Hank mentioned earlier the example of a BRCA1 mutation, which I think is a really great example because it's very helpful in thinking about the complexity of these issues. And I think this is a predisposition, genomic kind of situation. So if, for example, we were to decide to gene edit a female embryo, to get rid of that BRCA1 mutation or to change it back to the norm, it's an interesting decision if you think about it, because it's this situation with the BRCA1 mutation is not a definite causative mutation, so there's only an enhanced risk. And so in that kind of situation, I'm not entirely sure for myself if that would be an appropriate use. I can definitely see there would be potential benefit there, but the potential future female person that was created without that mutation, she may never have gotten cancer. And so we don't really know. We've just improved her odds. And so then you kind of get into this question, is there a slippery slope or not? And I'm not sure if there is or not. But then you can go to something like another gene that, say, if you change an A to a C, now we think you'll have a 40% reduced incidence of Alzheimer's disease. Should we go in there and flip that one? And then you can go, well, how about a 20% increased chance of prostate cancer or leukemia? And so for me, the dividing line between what's therapy and what maybe is not so clearly therapy, and this gets to like the ideal situation where we could comfortably use this, it's not so clear. I really think it is complicated.
Yeah, that was the example I was going to use, the BRCA1. But I'm not so sure that I agree with this worry that we're on this slope, that once we start there, we're going to end up at some bottom place that we can never recover from. I think we do a pretty good job of setting our boundaries on what we should do and shouldn't do, and I think that that is a moving target, and that discussions like this help us as a community of science and as a social community figure out where that boundary is going to go, whether it's just changing one gene for a predisposition, or perhaps not going there at all and only using this technology for in vitro types of research.
Go.
Well, sort of related to the BRCA, there are patients who in the past decided not to have children because they were worried about passing on the increased chance of their child having a disease. And so it has allowed, by being able to test for this, people to have children who otherwise may not have done it. And I think if you are able to reduce disease in your child, I think, that would allow more people to.
Louder.
Some risk, you can't really predict.
Welcome to life.
Let me take up the question of the BRCA gene. If someone is so concerned that they might consider not having children, then why not offer PGD or embryo screening for that?
Yes.
So for that particular case,
The only example, unless they're actually homozygous, if they've got a BRCA mutation on each chromosome, they would fall into this autosomal dominant homozygous case, although not fully penetrated.
But the other issue with PGD is that
which will not happen very often. Yeah.
there are situations where you test the embryos and they all have the mutation. So sometimes, you don't have normals to transfer.
Then you could make another batch.
But if you're older and don't get many embryos, you could go through multiple cycles.
Are there instances where you just have one embryo or maybe two embryos? A very small number.
Right.
Zero.
So sometimes you have very, very few. And, in situations where a woman is older and doesn't have many embryos, I think being able to correct abnormal embryos would definitely give them an improved chance of pregnancy.
Jump in there. So just to kind of build on that, in my lab we're trying to CRISPR different genes in cell lines. And we found it's called like simple and very precise, but it's actually really difficult.
Maybe you just don't do it well, Paul.
Yeah, I think we're screwing up somehow. But we work with millions of cells, so we can basically, this number issue kind of resonated with me. We can basically, one day we can try CRISPR on 1 million cells, and if it doesn't work out so well, but we still get 10 out of 1 million, for us that's a success. But I think if we implement this and we kind of transfer those odds, we're going to need a lot of embryos, I think, to get this to work properly. And so even in an instance like that, I think CRISPR will probably not be an option because it won't work in one embryo. You'll probably have to try 20. And maybe, if you think about that paper, even if we get way better than that, you're still probably only going to get a few embryos that have the right edit, they don't have the wrong edit, they look viable. And there's other issues, like we'll have to probably sacrifice some embryos to test to just see how well is the CRISPR working. And so I think I want to definitely dispel this notion that CRISPR is just sort of, one and done kind of thing. And I really think it's going to take a lot of embryos.
So it's both revolutionary like PCR, but it's not very good.
No, I think it is really good, but I think it depends on what your application is.
So I actually suspect if we ever went down the germline route, we probably wouldn't try to modify human embryos, but modify eggs and sperm or egg and sperm
Right.
progenitor cells.
Yeah,
Because then you could make 1 million sperm and only take the
right,
ones
and
that
there's
look good.
really exciting research going on with what are called human primordial germ cells, that you could edit those, 1 million of them, find some really great ones, and then make eggs and sperm from those, and then you're way ahead of the game.
I think Paul's comment goes right to this paper, and that's that this paper should have been published. I mean, what do you guys think? Especially, it's using a scarce resource, embryos, even though they were non-viable. A lot of this had been predicted in other papers. In fact, the Nature paper, I think, predicted all 3 of those results. And the scientists that have looked at this, the CRISPR scientists that have commented publicly on it, say that, well, it was kind of an obvious experiment. It didn't really move.
Yeah, but you never know whether something obvious is going to be true or not until you try.
You do it.
That's true. Other questions? Yes.
This
You
conversation was very much in the context of academia, and I'm curious, we've seen companies like about $100 billion funding. They're led by members who are on the IGI boards or anything like that.
One of them's led by the guy Shang from MIT, who's the other patent. There's going to be a wonderful patent. Lawyers are about to make $1 gazillion
There's a patent
because
dispute going
there's a
on right
patent
now.
fight
Yeah.
between the 2 groups that claim to have invented it.
My question is, how will discussion because we don't have
So I think, and we're already seeing it to some extent, I think industry is terrified of the germline stuff because that's just, political bad news for them. And in fact, the Nature paper, first author was a scientist from Sangamo.
First and second author.
Yeah, and a couple of more authors were from the Association for something medicine.
Regenerative medicine.
So these were all people from the somatic cell gene therapy industry, and that editor, I read that paper as saying, this is bad. This is not us. We're the good guys. They're the bad guys. Throw them into the pit. We're the good guys. Remember, we're the good guys. And I think we'll see that a lot. I mean, I do think that the industry interest will not be on germline gene therapy, which is going to be a relative, I think even in the wildest nightmares, will not be an enormous market, but it will be in the somatic cell gene therapy and it will be in the CRISPR critters.
And by the way, Hank, the Science article had 2 company founders on it too.
Yep.
George Church and Jennifer Doudna. Both of them have equity interests in companies, potentially large ones. So,
But
Yeah, I think that's
neither of whom
great point.
was sort of taking the same Manichaean, all good, all evil that the Nature paper did.
that's
I
true,
thought.
but still there were interests involved there.
And
think
I would
that's-
say that we have to think about the fertility industry in here too. That's a very lucrative, multi-billion-dollar industry. And the guy who developed the nuclear genome transfer, Shukat Metalopov, he started a company.
So specifically to your question, I'm editor at Nature Biotech, and it's been swallowed by industry.
Nature Biotech, which just had an editorial taking kind of a
That's true.
non-position on this. I
Yes.
couldn't figure out what exactly you guys were
I know
saying.
I guess the other thing is George Church is, at least from my perspective, he's much more enthusiastic about doing this in human beings than other people.
I was kind of surprised that George signed on to a paper that said any research
Yeah.
should have a
I
moratorium.
really thought it
but-
was surprising that he was part of that consensus paper because I interviewed him for my blog and he definitely talked about things like, well, this could be analogous to, giving your kid Ritalin or cleft palate surgery. So I really feel like George is trying to normalize this, and maybe he's not part of, that paper consensus in one way, but he was. So it is kind of surprising, and he has, his disclosure slide, if you've ever seen him give a talk on YouTube, has like 60 different companies' logos up on it. So I would definitely say let's see what George has to say about this too, because I think he is very enthusiastic about the commercial aspect.
I do think the fertility thing brings up an important point for me. If, as an advocate of this sort of regulatory approach, it would take, to get the kind of regulation that I think a lot of people would feel somewhat comfortable with, It would take a change in our current federal law. Because right now, once the FDA approves a drug or device or biological for one purpose, a doctor can use it for any other purpose. So you could not say now, we approve germline gene therapy, but only for homozygous, autosomal dominant diseases, et cetera.
Exactly, yeah.
Unless you change the law. And
Yeah,
the law can be changed.
But I think it could be very
And
politicized.
including sort of limiting the application by the fertility industry based on perhaps less than robust science saying we can regenerate your eggs this way.
Yeah, I would just weigh in a little bit. I'm a stem cell researcher, and the FDA has really been part of our discussion related to stem cell clinics offering treatments that are sort of on the edge or really technically against FDA policy. And the FDA has been really reluctant to take action on that. And so I do wonder if there was, say, a campaign focused on editing BRCA1 mutations, and that got sort of folded in with breast cancer advocacy,